he transferred or he came to IRI, and now he is looking after, I think, majorly in the wheat pathology. So welcome, sir, and floor is yours. Sir, student, they are all microbiology, entomology, and pathology students, and MS and PhD mix. So thank you, sir. Welcome.
Thank you, Dr. Suresh. Good afternoon to all. This training is basically genomics. Am I audible? Yes. Yeah. Genomics, it's big area of science, you can say nowadays. A lot of work has been going on in insect, pathogens or diseases also. Apart from genomics, I think genome sequencing, characterization, identification of genes which are related to virulence or susceptible genes. See, genome is big area of work. Apart from that, I will discuss how we can develop diagnosis markers for plant pathogens, especially fungus I will focus. Okay. So see, diseases are, so many fungal diseases are there. So fungal, why the requirement of diagnosis? See, have you seen any disease symptoms in field? Some are leaf spot, some are downy mildew, powdery mildew, some are complex. So we cannot identify. Based on symptoms, we cannot identify. So what other tools are required for diagnosis or detection of pathogen? That's why need of molecular biology. No? Basically, based on symptoms, sometimes we cannot identify the diseases. Either blast, either rust, sometimes confusing spot disease also. Beginning of that, based on that, we need to develop some molecule based markers to detect the fungi. In infected tissue also, on soil also you can identify. So, light, can you put off this light? Dr. Suresh, ये light कम कर सकते? ये light है ना, front light. Yeah. So, am I audible? I think without this also. Yeah. Mic. Yes. Yeah. Okay. Yeah, that is best.
ये
साइड से दिखाओ।
ये आप रख लो कहीं भी जहाँ कंफर्ट हो, इसमें भी आप लगा सकते हो।
ठीक है।
तो ये आपको अगर
इसमें माइक का वेरिएट करता है। ठीक है। Yeah. Am I audible now? Yeah. So what is the need of rapid and accurate diagnosis? See, like human pathology, similar way we need to identify rapid or accurate diagnosis, whether fusarium, whether puccinia, whether is alternaria, what exactly causing a disease. Okay, if you identified as a disease, then go for management practices. You can suggest a management practice to farmer. Ultimate aim is management, na? So diagnosis, that's why we require this diagnosis. Then I earlier also discussed symptoms. Symptoms, so many symptoms are there. If we discuss, you can see here different type of symptoms are produced by different plant diseases, either local, systemic or primary, secondary, microscopic or macroscopic symptoms are produced by different fungi. Some disease you can identify leaf spots like galls, canker, these are local symptoms you can identify. Systemic infections, wilting, yellowing and dwarfing, these are systemic infections. You know, I think little bit you know wilting disease based on soil-borne fusarium pathogens infect the root and causing the disease. As compared to foliar pathogens, the soil-borne pathogens are difficult to manage. Why? Because in soil the management practices are very few actually. How we can apply the chemicals, how we can apply the biocontrols for management of plant diseases. Then other you can see this club root of cabbage and root rot nematode. Nematode also a major problem, soil-borne pathogens. So among the major diseases which are important, you can see anthracnose, blight, canker, damping off, dieback, leaf spot, powdery mildew, rust, root rot, scab wilt in different many crops, either cereal crop, either vegetables, either oilseed crops, many crops it causing such type of diseases. So, while during identification of disease in field condition we collect the sample and visually we see the symptom. Sometime we will confuse whether it is a leaf spot or maybe blight or maybe damping off. Keeping in that view, we need to develop. So, some disease easily you can identify based on symptom also. Few examples, I am working on wheat actually. So, I have given few examples here. For example, one kernel bunt of wheat caused by Tilacia indica. It is, you can say, quarantine disease. Disease can be identified at the time of maturity. Again question comes, the infection already established in the year head, during maturity you can see the symptoms. It means we cannot diagnose in between during maturation of the seed actually. So, while harvesting of the seed, if you see these seeds are you can say hollow seeds, you converted these fungus Tilacia indica converted teleospores, mass of teleospores. These are teleospore, hard dark compact teleospore. While threshing it goes into soil. In soil it survives up to 5 years or maybe more than that. So, this is the source of primary inoculum. Okay. So, in next season it germinate and it reach to boot stage of wheat. You have seen wheat crop, I think maybe during January month the spore will germinate, that time temperature is low, it germinate and by monkey jump the spore will produce and cause the disease in the spike, and during threshing again it goes into soil. So, what the major source of inoculum is soil borne, soil borne. So, but in between how we manage this disease or how we can diagnose? Either we can directly wheat spike we can take, isolate the DNA and go for molecule tools, whether infection already present or not, that can be, we can do it. Other, for example, spot blotch of wheat, that is also major pathogen, particularly eastern part of India. You can see UP, West Bengal and then Bihar, where is warm humid conditions there. During Rabi season, the temperature 20-25 degree centigrade and high humidity more than 80%. So, it is basically it is soil borne. It is soil borne pathogen, it survives in the host stubbles or residue is there in field. So, the primary source of inoculum is basically Conidia actually. It is hemibiotic of pathogens, and during germination at the you can say 2-3 month old crop, it infect lower leaves of the plant, then reach to upper leaves. Basically, it hinders the photosynthesis. So, yield will be reduced, or by one important thing is that some fungus we can culture. Before going to molecular diagnosis, we can go for isolation. Isolation, any infected plant material you can take and isolate the fungus, go for morphological identification, either based on spore morphology or mycelial morphology. If you know, you can identify the fungus at isolation level. If not, based on spore, then we go for molecular tools. In bipolaris, we can identify based on spore morphology also and colony characters also, but sometimes we confuse because in Carvularia, Alternaria spores are same, more or less same. So, we have to do molecular tools for diagnosis. Like for example, some are biotrophic fungi, that fungi we cannot culture in artificial media, like these all are 3 rust, black, brown or yellow rust. So, these are all you can say, for example, pre-symptomatic stage you can diagnose, although you can see the symptoms in field condition, but for example, some latent infection is there. Understand my latent means before producing symptoms, before that you can identify such type of disease, because sometimes virus so you know, the pre-symptomatic stage you can identify viruses also like that based on molecular tools. Yeah. So, based on that brief introduction of this diseases, what are molecular tools we can diagnose the fungal pathogens? Basically, we are mostly following the PCR based methods. All the other methods are there, but PCR is best method. Why? I am tell later on. What markers PCR, real-time PCR, LM assays. These three are most commonly used by researchers. So, based on that you can develop the markers. I will tell you take unique, you can want to make unique sequence of primer, yeah, gene you can say. You compare similar species, for example ITS region. So, you take the ITS gene, take some part of gene and then compare to other you can say fungi where that type sequence is available or not, but you have to make uniqueness of that sequence that can amplify only that pathogen, not different pathogen. Like for example, we are target Tillisia indica. So, we have to generate Tillisia indica marker, it cannot amplify to Fusarium, Alternaria or Puccinia, it should not, then only you can say it is a unique marker. Okay. So, based on ITS, housekeeping genes, some housekeeping genes they commonly present plants also and fungi also, tubulin, elongation factor and genome based marker. Nowadays, see in NCBI you know so many genomes are available, Fusarium, Alternaria, Puccinia. So, based on you compare whole genome, you blast, you compare it, then you see any unique regions are there or not in a particular genome. Like for example, recently wheat blast in Bangladesh in 2016 it was reported. How they did? They directly take samples from the field and they did the genome sequence of this Magnafortia TTC and the sequence they blast. So, it is different than Magnafortia oryzae. They told that it is Magnafortia TTC, not oryzae. So, based on genome also you can compare the, or you can design new markers also. Yeah. PCR principle I think you know all, PCR you know, any doubt? So, it is very simple process. 3 steps, denaturation, aligning, extension you all know very well. Yeah. So, the important part in diagnosis is marker or your primer is important. How to design a primer, that is the important thing. What basis you are selecting, either gene based, either genome based you are targeting. That sequence should be unique. Before going to wet lab experiment, you analyze as bioedit or some softwares are there, it should not be match. In NCBI also you can blast. So, primer is important thing. Rest things you can do the PCR also, denaturation, aligning and extension, these simple steps are there. Yeah. See, PCR is best actually as compared to real time PCR. Why? Why PCR and real time difference you know? See, PCR we need agarose gel electrophoresis, okay, but real time there is no need of agarose gel electrophoresis. And in PCR we need more time as compared to real time, but here you can get good result also sometimes. See, here you can find some amplicon band in agarose gel, but real time you cannot see. Only based on computer reading you see where the curve is coming or not, melting curve is coming or not. Based on melting curve, the coronavirus you know, the marker is based on real time PCR. Do you know that? They allowed sometimes false reading also somebody has received, na? I am positive, I am negative. Why it has happened? Because some false reading also come. In PCR, maybe chances some false reading, but here you can see through agarose gel electrophoresis. The specific size of band should come and you see that is pathogen. Okay. So, sensitivity is important. Early stage of infection, how you can detect? Early stage, for example, some infected leaves are not showing symptoms. So, directly you isolate the DNA, plant also and pathogen both it will come, then do the PCR. So, early stage you can identify the pathogen is there or not. Okay. Rapid, so based on real time and PCR, PCR is not rapid actually as compared to real time is fast as compared to PCR. So, 24 hours is required, most whole day is required for PCR. If you isolate DNA, whole day is required, PCR and agarose all things is required. Yeah. Disadvantage what you say, during setting of PCR actually, maybe chances sometimes DNA is not mixed properly through pipette, some errors are coming sometimes. maybe primer may be sometime not added proper, not mixing, tech polymerase added or not properly. These things should be carefully added while you do the PCR. Important thing is required. Reagent is main, which reagent is most expensive here? Tech polymerase. In master mix is cheaper actually, but tech polymerase is costly. Yeah. The important thing is again primers actually. You design such type primers only amplified only those pathogens, not other pathogens. Why I am telling? Because if you are designing primers, sometimes they will amplify other pathogens also. That means your primer is not correct. So, primer designing is major focus to develop PCR based markers. Any doubt you can ask me in between also. Yeah, what PCR based we can do based on PCR actually in plant pathology. Overall I am discussing. One diagnosis, diagnosis and quantification. I will tell later on quantification also. Mating types also. Some pathogens have mating types. So, based on mating type genes, you can identify which mating type, like Phytophthora, A1, A2 mating types are there. If primers are there, you can identify which population which present where actually. Like hills, what population is there, in plains, what population is there. So, mating type also you can identify. Field surveys assess incidence. How incidence? Like for example, foliar blight, the spots are there. So, directly you can collect the samples and do the PCR. Which pathogen more as compared to Alternaria or Curvularia or Fusarium is there. So, based on that you can see how much incidence is coming. Distribution also you can tell. Quarantine programs. See how many pathogens are quarantined in India actually. Some are in abroad which are not present in India also many. Like Karnal bunt of wheat, it is quarantine pathogens. Till now it is reported in around 7 to 8 countries, not reported to other countries. So, while you can say export the wheat from India, they will check the seed lot whether pathogen is there or not. So, what they will do? Either infected grain they will take, either washing the seed and from washing material either water. No? So, that water from that they will isolate the DNA, and after DNA they will do the PCR. So, they can detect the surface contamination of the seed actually. Basically fungal, bacterial and viruses also, mostly more viruses are quarantine pathogens, but this wheat we have 100 million of ton metric ton of wheat we have in India, but export is major constant in Karnal bunt of, because of Karnal bunt of wheat. Because see is soil borne, during harvesting chances of infection is there. So, how to avoid that? So, major thing is that avoid or you can say detect the pathogen, which stage it can be reduced the infection, that is the major thing. Viruses, although diagnostic is major for viruses, as compared to fungal plant pathogens, more diagnostic protocols are available for the virus diagnostics. Why virus? Tomato leaf curl virus, New Delhi virus, Bangalore virus, Delhi virus, so many strains are there. There are more mutations are there, no? The virulence is more as compared to fungal. That is why the management in virus disease is less as compared to fungal and bacterial. Okay. Distribution also you can do virus distribution, identify alternate host, germplasm screening also possible. Although it is costly, if you develop some markers, for example, resistant genes, R gene based markers. But thing is, first you need to identify resistant sources, develop resistant markers, and based on that, for example, you are doing some genetic population. So, F1 population you take the plant DNA, then go for PCR, then see whether R genes are coming or not based on R genes. So, screening can be possible, but it is costly affair, it is not easy. A level of resistance also you can quantify the level of expression of particular gene. Yeah. So, flow chart how you can diagnose. First is sample collection. How any infected tissue you can take directly. Go for a DNA isolation, either based laboratory protocols or kits are available. Many kits are there. DNA isolation kit, fungal DNA, bacterial DNA, viral DNA kits are available. isolate the good quality DNA. Good quality DNA. Pure colony is a different thing actually. Only you find isolation of pure colony means isolation of fungal based on media, that can be also used. Then genomic DNA, then selection of molecular marker of PCR. What to do? Which marker you have to take? How? See, if you don't have any markers, see the literature first. Whether bipolar sorokiniana marker is available or not in literature, whether anybody has done work or not. Because we don't have all markers for all the pathogens, we don't have. Limited fungal pathogens we have markers. So if marker is not available, then what to do? Basic concept. The 16S or 18S ribosomal DNA, that is more or less present in fungal. Okay. That gene is already identified. Based on ITS region, you design, you synthesize the primers, then do the PCR. After doing PCR, what to do? The amplicon size should be, for example, 590 base pair. It should come like 590 base pair. Okay. Then go for a sequencing. Primer, connect PCR, then DNA fragment, then DNA sequence. Yeah, similar thing. So after PCR, check the amplicon size through agar gel, then go for a DNA sequencing analysis. You send for sequencing. If you are available in your institute, you can do yourself also sequencing. After getting the data of sequence, like 590 or 600 base pair, you got the sequence in FASTA file. So what to do? That FASTA file sequence you blast in NCBI, where it is coming maximum similarity, which fungal pathogen it is coming. Based on that, initially you can say it is bipolaris or fuserum, maximum similarity percentage you can see. Okay, is it clear? DNA isolation, PCR, sequencing, and sequencing, then NCBI blast analysis. Okay. So based on blast analysis, you can say this the bipolar sorokiniana pathogen is there. Understand my point? Very simple concept. In some cases, single gene is not sufficient to identify also. One gene sometimes not giving good result, like percent similarity 88% or 90%. Then we have to take other gene also, like tubulin gene or elongation factor may be recommended in literature, you can see also, because one gene is not sufficient to identify also sometimes. So you can take 2-3 genes which are most commonly present. Okay. That gene you have to take, again go for a PCR, then sequence, and all 3 gene based sequence analysis. Finally you can say this pathogen is this present in particular crop. Do you know plant disease general is there? Plant disease, you know plant disease, American Phytopathology Society is there. So there you can see every month hundreds of disease they are reporting actually, new disease they are reporting. So based on morphological identification, spore, then go for a PCR based identification. Both they combine together and then they are saying it is this pathogen is there in this crop. Is it clear? If markers is different thing, marker is based on these housekeeping genes, you can identify also fungal pathogens. Any doubt you can ask me? Okay. Yeah. So based on this, we have also done like that similar work also. For example, bipolar sorokiniana pathogen is there. This is the causing spot blotch of wheat. So based on morphology, we cannot identify much actually. So what we did actually, based on ITS 1 and ITS 4 primers are there. So we did PCR and we went for sequencing. After sequencing, and we done the blast analysis, and we generated the phylogeny also. See, whenever you make a phylogeny, for example, how we can make a phylogeny? These all are isolated pathogen you can see here, bipolar sorokiniana, different places, and you take the out group. Out group means, like if you plant pathologist, you know, I think, like Ascomyces fungi, you should nearby group of fungi. Basidiomyces nearby other fungi you have to take, whether they are sequence same or different actually. In this case, what we did actually, bipolar sorokiniana and bipolar oryzae we have taken out group. Oryzae means infecting rice actually. Sorokiniana infecting wheat actually. This is based on sequence we generated the tree actually, and other pathogens also, Spacifera also, Maedis also, bipolar Maedis infecting maize, bipolar Spacifera infecting wheat also. So, why we are taking similar type of genus? We can compare, we can say our bipolar Sorokiniana is clear cut showing it is bipolar Sorokiniana infecting wheat, same single cluster it is coming. Understand my point? Any doubt in this? So, always try to identify, take the other group also. So, you can say it is a bipolar Sorokiniana. And after that you can submit these sequence to NCBI. You have made your login ID, password, NCBI, have you made? So, after that sequence analysis you can submit all the sequence to NCBI, because your copyright, that sequences are your copyright, anybody can use, no problem, but after that you can get the accession also. Okay. So, it is better to submit in NCBI. Yeah, PCR is clear. Any doubt PCR based diagnosis? You can ask me. Same way, another technique is multiplex PCR. Nothing difference in multiplex PCR. It is a basically we can detect more than two. One based on PCR we identify one pathogen. On a multiplex we can identify more than two fungal plant pathogens. What we did here? You have to develop the 4 set of primers or 3 set of primer or 6 set of primers in single PCR reaction. Means you make a 4-5 primers, detect for example Alternaria, Fusarium, Bipolaris. So, you have to develop the 4-5 primers for individual pathogens and do the same PCR, but it is difficult to you can say standardize actually. Why? Same TM value sometimes it will not amplify. Melting temperature will be for example 55, but it should be ideal for 55 and all pathogens should be detected based on multiplex PCR. Basically viruses, so many viruses you can see here example also, so many multiplex PCR based markers are available. Fungus is very less in this multiplex PCR actually. So, basically this 4 type of strains, for example in flow chart you can see, here you can see we have take 4 primers, then for example 4 type of pathogens, 370 base pair, 275 base pair, 202 or 141 base pair. In same single PCR it is coming 4 by, it means 4 pathogens are there. Is clear? So, it is also easy, but the standardization is very difficult actually in this case. The same TM value all primers should work, otherwise it will not amplify. Yeah. So, any doubt in PCR, in fungal disease identification? Next step, recently the most commonly used technique, real time PCR. What is real time PCR? Have you seen? All you have seen I think real time PCR? Done practicals? Real time PCR. So, basically nothing it is. The amplification in real time you can say, whether within 2 hours how much amplification is going on or not. If amplification not there, you can stop the reaction in between also. In computer you can see whether amplification is coming or not, in between you can stop. That is why it is called real time amplification of particular gene. Understand my point? So, basically based on this fluorescent dye actually, cyber green we are using, and specific gene sequence also, probe also can be used for this real time PCR. Primer, master mix, this I will discuss later on. It also rapid and sensitive determination. Why rapid? As compared to PCR, it requires less time. And even sensitivity I will tell, maybe 100 femtogram, 1 picogram can be detected the DNA in the fungal infected tissue also. Apart from diagnostic, nowadays the most important thing is gene expression analysis. What is gene expression analysis? Transcriptome you heard lecture, transcriptome. So, any time point, any given time point, how gene is behaving expressed at different time point. For example, today this plant inoculated by some pathogen. So, what type variation is expression coming 12 hours, 24 hours, 48 hours. So, different time points how the gene is behaving, how much expression, how much fold is coming. If fold is higher, it means it is working, particular gene is working. Time point how gene is expressing, how fold change is coming. Basically, either defense side you can see, either virulence side also you can see. Any type genes, you according to your experiment you can decide which type of gene you are targeting for gene expression. Mostly in pathology we are doing host pathogen interaction type of genes, virulence genes, defense genes, R genes, all we are taking for gene expression analysis. Different kind of study. GMO, this different thing. Yeah. What is the basic flow chart of a real time PCR? What is that? First, we need to isolate RNA. Okay. RNA, then go for a cDNA synthesis. Kits are available. Okay. Then designing specific primers. Again, softwares are there. Primer 3 softwares are there. Design specific primers. Again, selection of primer is important here. Which gene you are targeting. Then go for a PCR reaction and quantification. Here is two type of PCR. You can see directly one step real time PCR and another two step. In one step, I think actually cDNA we are here making cDNA actually here two step. But here directly RNA plus RT reverse transcript we added. So we separately we are not developed the cDNA. So based on single reaction we are amplified the DNA, or here we are first we synthesize the cDNA and after that we set the real time PCR and then amplified DNA will be quantified. Okay. So what basic things are required? Water, cybergreen, primers, template DNA, this you can design. Why 10 microliter we kept? In PCR we are 25 microliter we are keeping, okay, because cybergreen is costly. So we are keeping less amount of reaction. The chemicals are very costly, be careful while using. The cybergreen I think one vial I think 25,000, 30,000 is available. So while doing, be carefully do the experiment, otherwise no result will found sometimes. So what advantage it has actually real time? Already we discussed monitoring reaction the valid course of avoiding risk of contamination. Means during reaction, during amplification you can see whether amplification is started or not. Okay. Rapid, no doubt rapid diagnosis of quarantine pathogens or any other pathogens, faster and more accurate detection as compared to PCR, require less reagent as compared to PCR. Here is two type PCR, then agro gel electrophoresis is required. Here is only PCR is required. Any doubt in between you can ask me. Okay. Directly amplification stage you can see whether amplification has done or not. Okay. Yeah. So in PCR one important thing in pathology diagnostics actually directly we can isolate DNA also. DNA also we can isolate and we design the specific primers for real time also, because real time PCR primers and PCR primers a little bit different. Why? In PCR mostly we can size is more than 300, 500, 600 we can take, but real time mostly we prefer less than 200 base pair. Why? Because easily can amplify. During amplification it can easily amplify. So for example, this is Telicia indica marker we developed based on qPCR, a real time PCR. So we designed some gene sequence ITS region and these primers we designed, amplicon size 150 base pair. Okay. This melting curve, that is very much important. While amplification, if you are getting such type of melting curve in the computer screen, that means amplification is going correct way, in right direction. If not coming like that, it means amplification not going. So you can stop the reaction in between. Okay. Another thing is CT value, threshold cycle value, you can calculate later on. This in computer can calculate. So, different quantity of DNA can be detected actually. What is that actually? See, what lowest dose we can detect. Like in this case, you can see up to 1 nanogram. If any infected tissue has 1 nanogram of DNA, that can be detected by the real time PCR. Again I am repeating, up to 1 nanogram of DNA is present in any tissue, any soil, so that can be amplified. But in PCR, maybe sometimes 1 femtogram or 10 femtogram, up to that we can detect. Means the DNA quantity, how much we can detect, that is the important thing. Yeah. Coming to specificity, what is the specificity? See, same primer, what we did actually based on cDNA, Tilletia indica, caries, Tritici, alternata, Sorokiniana, Puccinia and Chaetomium globosum. Up to 11, up to 10, these are all are Tilletia indica, others are other pathogens. It is clear that it is not amplification in other pathogens. You have to repeat again 2-3 times, be sure it should not apply other pathogens, otherwise it gives false markers also. Be sure while doing the PCR. Then sensitivity, what extent we can detect the DNA. So, here you can see up to 8 lane different concentration of DNA, 100 nanogram, 75 nanogram, 25, 10, 100, 1 picogram. Up to 1 picogram we can detect the DNA in the infected soil or infected leaf tissues. Okay. So, these are very much important. While publishing any paper, they will see what is the sensitivity of marker, what extent you can detect the DNA in the infected tissue. Is it clear? Yeah. So, what the important thing actually, what we did actually, under microscope, the teleospore in soil we analyze under microscope. Directly we have seen what the population of the teleospore. So, different samples we have collected from different places and we have seen how much teleospore per gram of soil. This under based on microscopy. Okay. So, 900 or 450 like that teleospores are present in the per gram of soil. What we thought we can detect, can we detect by qPCR? So, what we did here, from soil DNA we isolated directly from soil 1 gram 1 milligram soil we have done the DNA isolation, we did the PCR. Now, you can see here, for example, this here you can see 39,000 3 lakh I think maybe nahi, spores, femtogram DNA is there. No, just a minute, 1 minute, I want to see. Actually, I am showing, but how much teleospore we can detect? Here is there 100 femtogram. If the DNA quantity is 100 femtogram, during quantification, if the DNA quantity is 100 femtogram, it means it contains 14 teleospore in soil. We designed so many experiments, then only identified that how much teleospores can be detected in the minimum soil actually. So, if the quantity of 100 femtogram is coming, then we can say the 14 teleospore in the soil. So, that proved that the soils IRI field, like mostly the IRI field or kernel field. So, this much of teleospores you can compare how much this coming. If 100 femtogram 14 teleospore, then how much teleospores are there? Lakhs of teleospores are there, na? So, we proved that the soil is totally infested with the teleospores in the soil. The purpose was to detect the pathogen in the soil also and seed also. So, it proved that this marker working well and this can be detected the teleospore in the soil. In soil DNA means all microbes DNA will come, not one pathogen, other pathogens, microbe, beneficial, all DNA will come, but it amplify only Tilletia indica, others it will not amplify. Yeah. So, finished PCR and real time PCR we discussed. Now, recently new technology has emerged because some new techniques, new advantages also. loop mediated isothermal amplification, LAMP. It is very simple reaction as compared to PCR and real time PCR. Basically same 4 sets of primer we have to design. Online softwares are available, but need to select gene first. The 4 sets of primer you design it. LAMP means loop mediated isothermal, means this type of 3 prime 5 prime loop will be formed actually. So the product are the stem loop with several inverted repeats of target like structure within multiple loops actually basically, and amplification takes place 65 to 65 degree for 60 minutes. I will tell the methodology also. This is the basically based on DNA. Based on DNA directly you can detect, but initially it was developed for DNA, but later on the RT lamp means reverse transcriptase, means you can isolate the RNA also, make the cDNA, through cDNA you can make the lamp. Understand my point? Through RNA then cDNA can be used as a material for the lamp reactions also. That is called RT-PCR. The real, see those 2 terms are there, one is RT-PCR, one is real time PCR. Real time, so you know what is RT-PCR? Do not confuse your transcript. So same way the RT lamp you can say here. Okay. So for this actually for Thalassea indica we have developed also this marker Thalassea indica. 4 sets of primer we generated based on this software, the Primer Explorer V6 software, and after this primer we check in the NCBI blast any similarity is there not. It should not be any similarity to the other pathogens, that is clear cutting. Okay. These bioaudit all softwares are required for this analysis. Yeah, what actually this methodology actually LAMP. See, very simple technique. First is master thermobuffer is required, betaine is required, DNTP is required. Okay. Then magnesium sulfate is required, BST polymer is required, nucleophase water, HNB dye and primers. These 5 components is required for this reactions. So the protocol is this: first you have to take the master mix and then primer you have to add. Okay. It make to 25 microliter. Then keep mix for 95 degrees and 5 minutes. That is important thing, initial stage actually. Where how to do? In PCR machine you can do for 95 for 5 minutes actually, you know. Then enzyme C, BST polymerase should be added later on. After this reaction, this step you add the BST polymerase for 65 degrees for 60 minutes in heat blocks. In laboratory you have seen heat blocks are there. So that temperature should be maintained. After 1 hour, this HNB dye should be added, solution D should be added. So here you can see here, these are simple PCR tubes, up and drop tubes are there. Blue color indicates positive reactions, and this color is purple color means negative reactions. It means it is amplifying. Any doubt? Very simple process. Within you can say DNA is there now. The question is DNA. If any simple protocols are there for DNA isolation, then you can make a kit and develop, easily can be used by anybody also. Okay. So the basically it's very within you can say 1 hour, 2 hours you can finish this step. Or for example infected tissue based some easy methods for DNA isolation is required, easy methods. So if DNA is there, then directly you can do the process this one also. This is the kernel bundle. This also work with seed detection also we have done also, and soil also we have done. In soil also we have detected the Thalassea indica, and seed also we have detected. Okay. Yeah, same way you can see here also. Here also we have done the sensitivity also, different DNA concentration we have checked. You can see here up to this is the specificity you can see here. This only this one turns Thalassea indica, others are not amplifying, and sensitivity same way LAMP also we have checked the sensitivity, and this is actually 1 picogram here. Up to 1 picogram is here, up to fifth up to 1 picogram DNA can be detected based on the LAMP reactions. Okay. And this specificity also we have checked, this also other pathogens, with other pathogens we have checked. Clear? Yeah. Number of publications are there. You see here which genes are there: Phytophthora, Verticillium, Botrytis, Fusarium, Puccinia. All markers has been developed based on the LAMP assay. LAMP assay. What genes they targeted? You can see here: elongation scar marker, RAPD, ITS, tubulin, 6 gene ITS. Different type of genes they have taken and they developed the primers. The story is only primers. If primer is correct, you design properly, then it is working well. What primers you have taken, that it depends on amplification of the any target pathogen. So that is part of PCR, real time and LAMP. 3 majorly we are using nowadays. Genomics, a little bit I will discuss the genomics also. We have also sequenced genome based on Illumina and PacBio also, and this paper also published, you can see also. But see, genomes in fungal, so many genomes are available. But the question comes, all are draft in nature, not completely sequenced. Completely sequenced based on chromosome based assembly is not present. So if you are not completely sequenced, then where to target or which gene we can target for the gene silencing or genome editing actually. See, one pathogen has in fact causing disease in plants. It has so many pathogenic genes related genes, like virulence genes, toxin related genes, chitinase genes, so many genes are there. So which genes can be major role in virulence? Can you identify? The question comes, so many diversity also in fungal pathogens, mutation, recombination, so many things are there. So if variation is coming, which gene can be targeted for any knockout or transgenics? Because the population or any pathotypes are there, fungal pathogens, they are changing day by day also. For example, we collected sample this year. If you isolate next year, the pathogenic level will be different actually. You can say virulence nature of pathogens are changing because host pathogen environment interaction is there always. So they are trying to evolve new pathotypes. Okay. This is also available in NCBI. One important technique, just we are started this work on that actually. What we did actually, for example, we have selected 4 genomes: Indica, Caries, Walkeri and Lewis. We selected 4 similar species. So we tried to develop these markers based on genome actually. That we have to do PCR also, but based on genome comparison. Genomes of fungal 30 to 40 MB, mostly 30 to 40 million bases are there. So based on genome and bioinformatics tools, you can design the primers also, genome based also. Because nowadays NGS technology are cheaper as compared to earlier also, nowadays cheaper. If for example, one pathogen genome sequence maybe based on Illumina maybe 1 lakh rupees. Any pathogen, if you are using Illumina and PacBio platform sequencing, maybe 2 lakh or 2.5 lakh rupees it is required for genome sequencing. So that is also genome based, so many things we can give answer also. Like see, during interaction of pathogen, what type genes are coming, what actually having role in virulence, how further we can address those questions to management, what should be targeted for management any pathogens. So another study also we have done genome wide. So GWAS, GWAS technology you have to. In plant science, GWAS is mostly used, for trait specific QTLs they are identified. But in fungal cases, what we did actually, we have collected population of fungal Tillecia indica. So we did the GWAS and we identified some virulence genes. These SNPs related to virulence in 13 positions actually. So they are targeting trait specific QTLs. Here we targeted if some virulence loci which having role in virulence. So what we do, if you know the virulence loci, the corresponding gene in the host can be identified. Again I am repeating, if you know virulence loci in pathogen, okay. So these corresponding genes in the host can be identified? You can say S gene, S gene. That S gene can be silenced or not? Now genome editing is nowadays big way it is coming, na? So that S gene can be silenced. But the question comes, we need to identify S genes. You don't have much S genes in plant fungal pathogens. Lot of variation is there, lot of gene clusters are there. So that the questions identify single gene which are having major role in virulence. So many genes are having role in virulence. Target single gene, that is the question actually. These are different papers aspect we have done actually. Genome also, expression analysis also, then marker transcriptome also, real time PCR marker also we have done, then functional part also we have done based on this work actually. Thank you. Any questions you can ask me now. Do you understood? Basic concept actually, not detailed methodology, you can read it no problem, but concept should be clear. Concept is very much important. Any questions, any doubt, you can ask me. Please ask. All are PhD students? No question? Already understood, I think. Completely understood, I think. क्यों रजना? Okay. Basic things. Otherwise, I will ask also. Otherwise, I will ask the question. So, is the diagnostic important tool? What type disease is important? Which type of disease is better for useful? Foliar, seed-borne. The more important, latent infection is more important. Because see, if symptom already producing by some fungi, na, so easily we can identify these very. Okay, but sometimes if not producing a symptom, there is a need of diagnostic. Like in kernel burn, some hidden symptoms are there, Puccinia also. So there is more important diagnostics. Export import do. Quarantine centers, it's more useful. While export import the material, they will use the diagnostic marker. Easily they can identify, detect the pathogens. Quarantine centers. Farmers will not use diagnostics. Only private companies, those in virus diagnostics also, bacterial diagnostics and fungal diagnostics. So private player, they will use the diagnostic protocols. To import the material, to export the material, it is very much important. I think no questions, I think.
So, because sir was here with us for the past 1 hour,
Okay.
and each of the lecturers who are having here is having ample experience, and they are giving time to you, and they are explaining the things to you, and you know how many people are sharing their experiences with you. So Malkhan sir, abhi यहाँ पे I think sir this will be next year will be your 10th year in IRA, right?
Yeah,
You
yeah,
came in
10th.
2015. Sir came here in 2015. I also came here in 2015.
Yeah,
But
nice.
sir came with a lot of experience from different institute, and so first IRC I met sir in one of the IRCs, because crop protection IRC is together. So sir presented the work of that institute here. That's why it means like we will remember somebody in something, no? So I was thinking, sir क्यों present करे, मतलब उधर का काम इधर दिखाने की जरूरत नहीं है, फिर भी वो कुछ the level of sincerity and all, it counts. Whenever we go, at least nobody should think that we haven't done anything, those kind of things. So Malkhan sir का ample experience है, whatever he has taught, he has talked to you, or all from his experiences, and sir का knowledge आपके साथ share किए. So on behalf of you all, and on behalf of the organizing people of this whole training program, I would like to present sir a token of love for his valuable time with us. Thank you, sir.
Thank you so much.
मैं फिल्म से निकल आई, मैं फोटो नहीं लेना अच्छा है. Thank you, sir.
Yeah, thank you.
हाँ, मैं बुला लेती हूँ.
उनको बोल ऑनलाइन भी.
Okay. Oh, today there is an end of the session, so the online candidates can also log off. Okay. Half an hour early we are leaving you all.